Hello, we're going to talk about enzymes today and the reactions that they help catalyze. So remember, enzymes are proteins. That means they're made of amino acids and Sean. So some things about chemical or biochemical reactions. So a biochemical reaction is when there's a change, some type of chemical substance into another. The key term of reactant is what starts the reaction, and a product is the result of that reaction. It's formed from the reactant, and we represent a chemical reaction through a chemical equation. So you can see the example there of methane plus oxygen gas releases carbon dioxide and water. So that is an example of a combustion reaction because carbon dioxide and water are products of the reactants of methane gas and oxygen gas. One thing we have to remember with chemical reactions is that law of conservation of matter that we looked at back in our ecology unit. Remember, matter cannot be created and restored but transformed. And so with conservation of matter within chemical reactions, we have the same number of each element in the reactants in the products, and matter is always conserved. So if we wanted to count atoms, we would have just one carbon atom as a reactant in a product. We'd have 4 hydrogens. We would have 4 oxygens, and it's balanced on both sides. And again, that is because matter is conserved during a reaction. Looking at reaction types, there are endothermic and exothermic reactions. Exothermic is when energy is going to be released, and endothermic is when energy is going to be absorbed. So another, remember, we talked about with ecology, not only is matter conserved, but energy is also conserved during chemical reactions. So the total energy of the system is the same before as it was after the reaction. And like I said earlier, exothermic reactions release energy, usually in the form of heat, and endothermic reactions absorb energy. So some examples of exothermic reactions include the combustion of fireworks, and also freezing is an exothermic reaction because heat is released in that process. Examples of endothermic reactions, opposite of freezing, so melting of ice, and the boiling of a substance are also examples of endothermic because those require an input of energy. Now looking into our bulk of our lesson on enzymes, remember there are some things we already know about enzymes. 1 is that they help speed up reactions. 2 is that they're proteins. And 3 is that they're not consumed by the reaction. So again, some things to remember that we should already know. Proteins are made of amino acids. We know there are 20 types of amino acids. And what varies in those 20 types is they are a random group. Amino acids can be arranged in various ways, leading to a limitless variety of proteins. Proteins have very specific functions within organisms. Enzymes are a type of protein. And the protein function depends upon its structure. So remember those 4 levels of protein structure: primary, secondary, tertiary, and quaternary. Without a protein structure, it would not be able to function. So we're going to look at some characteristics of enzymes. Remember, they are proteins. They're catalysts, so they're going to speed up reactions. They're reusable, which means the enzyme can perform the reaction multiple times. They're substrate specific, and they're also sensitive to changes in temperature and pH. So enzymes like a very specific environment, and if that environment changes, then they may not function. Let's add in some amino acids there to our polypeptide chain. So the overall general function of an enzyme is to help control metabolism. And remember back from chapter 1, our first chapter, metabolism is just the sum of chemical reactions that take place in an organism. And these chemical reactions, these metabolic reactions, can be controlled and sped up or accelerated by the use of enzymes. Most reactions that occur within organisms would occur too slowly if it wasn't for the presence of enzymes. Most enzymes end in ase. That suffix of ase is like we saw with sugars, ose. Enzymes mainly end in ase. So that's a little hint there. All reactions need like a little push to get started, and that's where the term activation energy comes into play. So all chemical reactions need energy to start the reaction, and activation energy is the energy needed to start the reaction. So what enzymes do is help lower the amount of activation energy needed to start the reaction. So again, enzymes are going to help speed up or accelerate chemical reactions. And like I just said, they're going to help lower or reduce the amount of activation energy needed to start a reaction. So you can see this graphic here, and there are two reactions, and one is with an enzyme, the blue curve, and one is without an enzyme, the red curve. And you can see that with the enzyme, the activation energy is much lower compared to that without an enzyme. So this reaction in blue would occur much faster than the reaction in red. And this is showing respiration. We have our products of glucose and oxygen, and reactants of glucose and oxygen, and products of carbon dioxide and water. So how do enzymes actually work? Well, each enzyme in structure has what's called an active site. And you can see in that model there, that little animation, the active site is where the substrate is going to bind to the enzyme. And what a substrate is just simply a reactant. So I'm going to show that again. So the yellow is pretending to be the model of our enzyme, and the kind of grayish green and the bluish shapes are the reactants or substrates that come together and bind in the active site. And that's where the reaction is going to take place specifically, is in that active site. So enzymes can work two ways. They can help synthesize or produce products, or they can help break things down. So this is an example of a synthesis reaction. So we have two individual substrates fitting into the active site, and it forms what's called the enzyme substrate complex. That's converted into product, and then it's released. And notice the shape of the enzyme does not change, and it's also ready to perform more reaction. The opposite here is a decomposition reaction, which means it's going to break things down. So that reactant, that substrate, it would actually bind a whole lot better than that. It would bind perfectly into that active site. Again, form the enzyme substrate complex. And then those two pieces are going to be broken down since it's a decomposition reaction, and then released. And again, the shape of the enzyme did not change, and so it is reusable. So this is called the lock and key hypothesis. One substrate for specific enzymes. Enzymes are very specific. They will not act on isomers. There is one substrate for each enzyme. So that substrate analogy there as a lock and key, in this substrate acting as the key fits that lock, but it will not fit another lock, or another key cannot fit that same lock. So even with isomers, like we talked about, same formula but different structure, like glucose and fructose, they have different enzymes that act upon them. Even though they are structurally different, but chemical formula is the same, they can't use the same enzyme because their shapes don't fit the active sites. So again, one enzyme for every substrate. And there's a very unique fit with that substrate and that active site on the enzyme. And that's called the lock and key hypothesis or lock and key model. One of the characteristics again about enzymes is that they remain unchanged, so before and after the chemical reaction, and they're also not used up during the chemical reaction. That's what makes them catalysts. So the reaction ends when there's no more substrate to convert to product. And again, we looked at this earlier, but there can be catabolic reactions, which would be a synthesis reaction. That would be a decomposition reaction. Or there can be anabolic reactions where there are synthesis reactions. So catabolic is the same thing as decomposition, and anabolic is the same as synthesis. So again, these are the same before and after the reaction. Now lastly, looking at environment, each enzyme is very specific about its environment. The one thing they're very specific about is temperature. Most enzymes work best at body temperature. Remember, we are science, so we want metric. So that's around 37 degrees Celsius, not Fahrenheit. So we can look at this graph here, and if we didn't know that, we can see that green X there, optimal. Optimal means like ideal or best. So most enzymes, if we found that green X at the top of the curve, or a peak, and came down, it would be around 37 degrees Celsius. Most enzymes are going to what's called denature if they lose their optimal temperature. And so what that means, denature, is that they lose their shape. And remember, shape is very important for enzymes. So if they lose their shape, they can't bind substrate. That active site no longer will bind substrate, and so they can't function. And so notice enzymes will denature, they'll lose their shape at very high temperatures, and they'll just go inactive at low temperatures. So inactive means that they aren't losing their shape, they're just not going to work. A little effects here if we were in person. And the same thing with pH or acidity or basic environments. So most enzymes, again, want an ideal or optimal pH also to function at. So we have this graph here, and it says what pH does pepsin work best at? So notice pepsin is located in the stomach. So it is going to function best at a pH of 2, which is very acidic. It is found in the stomach, so it's an enzyme that helps in digestion. So that makes sense. We have a second question here of what pH does pancreatic lipase work? Notice that ACE of lipase, that tells us it's an enzyme. This is going to be an enzyme found in the pancreas. Notice it's the opposite of pepsin. It wants to work best at a pH of 10. So it works better at a basic pH. But most enzymes, you can see, this is their optimal curve here. So if it came down, it'd be around a 7, which is a neutral pH. So a pH of a substrate higher or lower than optimum pH, it can denature that enzyme, making it ineffective. So then again, its shape would change. It would not be able to bind substrate, and it would not function. So shape is super important when it comes to enzymes. And again, optimum pH, same thing as optimal temperature, is the pH or temperature value at which the enzyme has the highest activity, which means it has the fastest rate of reaction, which means it can produce the most product. And again, different enzymes can have different optimal pHs, same thing with temperature. We can see that from this graphic that we have 2 different enzymes here, and they function best at different pHs. So that wraps up enzymes, and we can talk more about enzymes on Wednesday, but make sure you got the notes ready to go on enzymes for Wednesday. Bye.